All right, guys, we are going to begin our final unit today. And we've already talked a lot about plants and we talked about taxonomy. We went in detail about their leaves and their stems and their roots. And we talked about how things are transported within plants. So we've kind of went through just about every topic we can, except for how do we use plants? Maybe if you're going to school for some type of botany, you might be interested in forensic botany. So we're going to look at that as our final unit. We'll be spending a couple weeks on it. And the very last week you'll have to dedicate to your leaf project, which I explained a while back. So forensic botany, the story that plants tell all. How are plants useful in forensics? We can look at how seeds and spores can attach to clothes. Pollen is going to be inhaled into nasal cavities. Diatoms are found in the lungs of drowning victims. Vegetables can be found in the stomach contents of victims, and lichens may be found in the cuffs of suspects' pants. So as I go through these, you're going to want to have open the notes PDF so that you can take your notes in those boxes and fill all that out, because you will be turning those into me at the end of this. So all of those things can give us clues as to where the person was or what they were doing whenever they died. Perhaps somebody was killed and then they were thrown into a river. Well, we would know that they were not in fact drowned, but that they were killed prior to that. Or those diatoms, if they did drown in the river, that might give us a hint as to what river they died in, if they weren't pulled out of it. So what is forensic botany? Forensic botany is the application of plant science to crime scene analysis for use in legal cases. It can be used in civil cases to determine who's responsible for root damage to sewer lines, livestock that have been poisoned by non-native plants, or landscaping contract disputes, just as a few examples. The following questions may be answered through the study and use of botanical evidence. So who was the victim? Who was with the victim or at the crime scene? Who were they at? Wow. Who was with the victim or at the crime scene near their time of death? What did the victim eat before dying? Plants tend to stay in your stomach a little bit longer because it's very hard for people to digest cellulose. When was the crime committed? And when was a specific person, vehicle, or article at a crime scene? So it might not tell you specific times, but more like seasonal changes. How long has it been since the victim died? The post-mortem interval, the PMI, and how long was the body buried? The first mention in history of forensic botany was in Plato's Phaedo of 399 BC, and it describes Socrates' self-administered death sentence of poison hemlock. Legal acceptance of forensic botany as evidence first occurred as a result of evidence presented during the trial of Richard Hauptmann, the man who kidnapped and murdered Charles Lindbergh's son. Forensic botany became part of the training for FBI agents at the National Academy in Quantico, Virginia in 1993. To successfully solve cases, one must first understand plant ecology and how plants interact with other plants and its environment. Plants grow in assemblages, which is groups of plants usually dominated by one species. Assemblages share the same habitat requirements such as the soil type, moisture, sunlight. Maybe it's the wind, their altitude, and their latitude and longitude. So you're going to find certain plants growing in only specific areas. A forensic botanist may need to explore various locations to fully explore and investigate a crime. They may have to go into a forest, a desert, a tropical rainforest, a pond, lake, street, inside a home or a car. all of those places. Gastric contents and forensic botany. What parts of a meal can withstand the action of the digestive juices, and how will the gastric contents help to solve a crime? Plant cells with strong cell walls are made of cellulose, and they can easily withstand digestion. These plant cells in a person's last meal can be used to estimate post-mortem intervals based on the degree of digestion. Knowing where the victim had been prior to death helps the investigators because they know where to start questioning people who last saw the victim. Now, obviously, you're not going to be pulling a salad out of somebody's body and say, oh, that came from Applebee's. We'd better go back to Applebee's and start questioning people. But at least you would know that they were someplace where there was a salad. After examining the crime scene in full, the forensic botanist follows all procedures for evidence collection, documentation, photography, recording, and maintaining a chain of custody. A general description of the dominant plants should be included, along with notations on the presence of broken branches or disturbed plants. So whenever they actually go to the location, they'll take a look and write down all of those things there. If something was broken or if they saw a lot of shoe prints, maybe there was some type of a fight, and that would help them to understand maybe what happened. So how much should you sample, and how should you package and label your sample? In the case of botanical evidence, the recorder should enter the following information on the label. Description of the plant, such as the tree, the shrub, the vine, pollen, flower, is it woody or herbaceous, or is it algae? The height of the plant if the entire plant was not collected. Color and shape of flowers, fruits, seeds, stems, and leaves. While it is beneficial to have multiple people searching for evidence, only one person should actually do the collecting to avoid duplication of specimens. So they'll be writing down everything that they collect, and that way you'll know that you just have one specific sample, so you're not testing things over and over again and duplicating it. You want to collect at least 10 different types of plants from the area's assemblage, and this is called a habitat sample. You always want to place botanical evidence in paper, never in plastic. And you never want to freeze botanical evidence. And we know these things aren't going to last forever, so you do have to go through and take a look at your samples fairly quickly. General guidelines for collection of different types of plants include the following. For woody plants, you want to collect at least 8 to 12 inches of the plant showing several leaves with variation in size, color, and leaf arrangement. So you don't just want one leaf off of a plant, you need multiple. Tall plants or vines should be zigzagged, not coiled in paper. Small plants and fragments should be stored in large pieces of newspaper. Broken branches should be cut at least an inch above and below the broken area. So you want to see how that branch was cracked. And if you can, you want to collect the roots when possible. Now, some of these things you may not be able to do depending on your location, but you want to do it to the best of your ability. Fruits can be sliced and placed in thick newsprint or between sheets of wax paper if the fruit has a high sugar content. Larger dry items, such as pine cones, can be placed in a box or a larger paper bag with the bottom flap closed. Pollen versus spores. The male reproductive structure of a seed plant is called the pollen grain, as it is as small as a pinpoint. A spore is the reproductive structure of algae, some plants, and fungi. The scientist who studies palynology is interested in the field of pollen and spores. Both pollen and spores have burrs or hooks with jagged edges that enable them to easily attach to hair, blankets, and clothing. Cellulose, the sugar that comprises the cell wall in plants, chitin, makes up the cell wall in fungi, is very durable and it persists for a long time. For this reason, pollen grains can last for decades under some conditions, making them very useful in forensic investigations. Seeds They can be packaged and they do last for years. They won't start to germinate until the conditions are correct. Pollen spores have different functions, but they have similar characteristics, such as being microscopic and having a resilient cell wall. The use of pollen and spores in forensic investigation is based on Locard's principle of exchange, which states that every contact between two objects or persons leaves a trace. Even if you don't think it's leaving a trace, you're leaving a microscopic one. So what are some useful ways that we can use pollen and spores in an investigation? We can locate a crime scene or a grave site, and we're not talking about, the graveyard that they're buried in, but the site where the person was buried by somebody who didn't want them to be found. You can determine if a body was moved. So if there are pollen or there's spore pieces on them, you'll know that if it's not from that area, obviously that body was killed somewhere else. Link a suspect to a crime scene or victim. You might be able to find spores on somebody's clothes that they threw in a dumpster. And confirm or refute an alibi. By understanding pollen production patterns for plants in a given location, one can either predict the type of pollen fingerprints or pollen profile to expect in samples. That is, the number of type of pollen grains found in geographic area at that time of year. So that is what we're looking at. We're looking at seasonal changes. This is just a graph here showing you the pollen profile. And these are all different types of trees and where they would be located at. The plant kingdom can be classified into two groups based on how they reproduce: non-seed plants and seed plants, which we have talked about before. The earliest plants were non-seed plants and reproduced by dispersing spores. They include the ferns, the mosses, the liverworts, the horsetails, and the club mosses. Some of the more recently evolved seed plants also produce spores, but their primary means of reproduction is dispersal by seed. So they actually have two ways that they can go about throwing their seeds out there. So for the seed-bearing plants, during their life, seed plants produce cones or flowers that make pollen to disperse the male gametes. Seed plants existing today include the gymnosperms and the angiosperms. We're talking about the cone guys versus the flower guys. Seed plants are the predominant land plants, so they are the most likely plants to leave evidence at a crime scene. Gymnosperms are the oldest seed plants. They're exposed and are not enclosed in a protective organ like the fruit. They include the cycads, the ginkgos, and the conifers, which are your cone-bearing plants. Conifers are the largest, most familiar group and include pines, spruces, firs, junipers, and other evergreen plants. Many produce their seeds within a hard structure called a cone. There are female and male cones, of which the female cone is typically larger than the male cone. Inside the female cone are ovules, while the male cones release large amounts of pollen grains. Pollination takes place when the pollen produced by a male cone is spread by wind currents to the female cones. Sperm cells from the pollen grains fertilize the eggs inside the ovules, and the seed develops from a fertilized ovule. Angiosperms are the flowering plants, and they produce seeds within an organ called a fruit. Angiosperms are very diverse, and they include corn, oaks, maples, and the grasses. There are about 300,000 known species of angiosperms, and they are found in almost all habitats. Due to their diverse habitats, angiosperms are found at most crime scenes. The basic reproductive unit of an angiosperm is the flower. The pistil is the female part of the flower that produces eggs, and the stigma is the part of the pistil where pollen lands. A pollen tube produced by a pollen grain goes down the long, thin style until it reaches the ovary. The male part of the flower, the stamen, is responsible for pollen production. The long, thin filament of the stamen elevates the anther and produces pollen sacs. Following pollination, one of the released sperm cells successfully fuses with an egg. The ovule becomes a seed, and the ovary develops into a fruit. There's two types of pollination, and we would have talked about these probably for the first time when you were in elementary school, and then you definitely hit them again in biology. So cross-pollination involves two distinct plants and is more probative in forensic investigations because it would narrow a field of search to a more specific area due to cross-pollinating plants producing more pollen than those who self-pollinate. A self-pollination is going to be the transfer of pollen from an anther to a stigma on the same flower. And that's how Mendel did his pea plant studies. So cross-pollination, you have two completely different plants. Self-pollination is on the same plant. Spore producers. Spores are asexually reproductive structures that produce a variety of organisms, including algae, fungi, and plants. Spores are dispersed in a variety of ways. Algae are going to release their spores directly into the water or the air. Fungi depend on wind or water dispersal, depending on the species and where they're located. And ferns and mosses, which are your seedless land plants, they release their spores into structures called sori, or sporangia. Bacterial spores, which are the exception. When environmental conditions are unfavorable, some bacteria form thick-walled resistant spores called endospores. Endospores are different from the spores produced by fungi, algae, and plants because, for one, endospores are not used in reproduction, and two, a bacterium can produce only one endospore at a time. Endospores are of interest in forensics because several types of bacteria that make endospores can cause diseases, such as anthrax and botulism. So using these things as evidence, when viewed under a microscope, the hard outer layer of a pollen grain or spore has a unique complex structure. It has distinguishing features that include size, shape, wall thickness, and surface textures, such as spines. And we're talking a lot about spines right now when we're dealing with the coronavirus. Even though we're talking more about proteins, it's still a little spike that comes out of the cell. So these are all different types of pollen and spores, and you can see that they do have a wide variety and wide range of sizes and colors and shapes. So, for example, larger pollen grains, such as that of corn, cannot travel far and can only drift with the wind about half a mile. So someone with corn pollen on him or her has probably been close to a cornfield or a cornflower. So these botanists, they know how far different pollen spores can travel, and that helps to limit their areas down for what they're looking at. It is critical that forensic palynologists carefully and methodically collect and store samples while avoiding contamination. Contamination would result in the evidence being considered admissible at court. Pollen and spore evidence can typically be found in the following locations at crime scenes. So it's a lot of locations. So we're looking at living and decaying plant material, soil, dirt, mud, and dust, hair, fur, feathers, clothing, shoes, blankets, rugs, baskets, carpet, and rope. Victims' hair, their skin, their nails, the respiratory tract is a big one, their digestive tract, and fecal matter, honey and other foods, paper, money, and packaging materials, vehicles including tires, windshields, air filters, and undercarriages, furniture, air filters of homes and airplanes, cracks and crevices in floors, walls, roofs, and fences, and drug wrappings or containers. So there are a lot of potential places to find spores. When you're collecting them, all samples are collected by a forensic palynologist wearing gloves and using clean tools, such as paintbrushes and cellophane tape. Each sampling instrument is cleaned after each sample is taken, or a new one is used each time. All samples should be placed into new, dry, sterile containers, such as paper bags or paper envelopes, and then dried to prevent decomposition. After the pollen and spores are processed and chemically extracted from the samples in the laboratory, forensic palynologists examine them using microscopes. They are best viewed with transmitted lights or phase contrast microscopes. Additional details of surface features may require the use of a scanning electron microscope. So let's look at a couple case studies. Our first one is on Natalie Mirabel, and you will be including in your own notes here how forensic botany played a role in solving their case. So you're providing just an overview of what I'm talking about here. Dr. Jane Bach, a botanist turned forensic scientist, was called to investigate the murder of Natalie Mirabal in 1999. Her body was found in a canyon off a road leading up into the Rocky Mountains in Colorado. Her husband Matthew had an alibi that he was home with their daughter. Botanical evidence found in his car's undercarriage, floor mats, and pedals showed that Matthew's car had recently been in an area with plants growing in higher elevations, like those found in the canyon, and not the type of plants found growing near their home, so they didn't live in the mountains. A combination of motive, a new life insurance policy, and multiple forms of circumstantial evidence, botanical, the wife's blood on his gloves, his DNA under the wife's fingernails, led to his conviction for his wife's murder. So plants did play a big role in that. They also used, the DNA. She must have tried to scratch him because obviously she didn't want to die. Who would want to leave their new baby at home with a crazy person? The next case study is on Samantha Forbes. Samantha Forbes was seen leaving a bar with two men while a tropical storm passed over the city of Freeport, Bahamas in 1999. Her body was found the next morning on a local golf course, but little evidence was recovered at the crime scene due to the storm. Police did recover a tiny fragment of a blade of grass from one of the suspect's socks. Dr. Bach was able to identify the grass as almond Bermuda, a grass that was specifically selected for that golf course. The two suspects were convicted based largely on this evidence, and because no other golf course on the island used this type of grass. So just a piece of a blade of grass. So they really take their time on looking over their bodies. The next case study is on the Goldhead Branch murder, which is in 1991. Forensic botanist Dr. David Hall got his first clue in the case when a 5-inch turkey oak seedling was found growing through the human tibia bone, where the bone had rested on the leaf. A line of dead cells matching the size and shape of the human bone was visible. Without sunlight, leaf cells cannot produce chlorophyll and they die. Dr. Hall estimated the time it took for the shading to kill the leaves. This produced the first time interval. The leaf had no malformations, which indicated that the bone was dropped on the leaf after the leaf was formed. Dr. Hall estimated when budding occurred and thus was provided with his second time interval. It was determined that bite marks on the bone matched a large dog, and a third interval was calculated based on when decomposition would have reached a point that the dog could separate the bone from the rest of the remains. Adding the time intervals together and working backwards, the result placed a deposit of the remains during a cold winter, which would slow down decomposition of the body. The skeleton remains were found to be consistent with a missing persons report from a week before Dr. Hall's estimate. So that shows you how they can pinpoint different times. He got it awfully close to whenever that person died. Drowning victims. Now, at the very beginning, we mentioned diatoms, and diatoms are relatives of algae. And I know how much you guys enjoyed algae from our first study, which was about 2 weeks long of just algae. They have cell walls containing silicon dioxide or silica. They have distinct shapes that are easily identified under a light microscope. When a person drowns, he or she inhales water deep into their lungs and body tissue. Algae and diatoms are non-vascular photosynthetic organisms. They're important producers found in freshwater, saltwater, and mud. By comparing the number and proportion of algae and diatoms in the lungs and tissues, it is sometimes possible to determine where he or she drowned. So the pond attack. 2 young brothers were attacked while fishing in a pond in Connecticut. The assailant beat both boys with a baseball bat, bound them with duct tape, and left them to drown. One boy escaped and helped his friend. Mud found on the boys' sneakers contained the same diatoms and algae that were found in the pond. When a suspect was apprehended, police collected mud from his sneaker and found that they too had algae and diatoms similar to those found at the crime scene. The most convincing evidence that linked the suspect to the crime scene was the proportion of different forms of algae and diatoms was consistent in all 3 samples. So they collected samples from the 2 suspects, or from the 1 suspect and the 2 victims, and they were able to link them all together. Secrets from a grave. A series of kidnappings has occurred. Several victims' remains have been recovered from a shallow grave. When another person in the area goes missing, the police search for a missing person and for evidence of a recent grave. So how would you recognize a grave site? If it is an old one, then the actual site sinks into and fills the grave as the ground settles, so it's not nice and level anymore. If the grave site is new, a mound of soil will be visible near the grave, perhaps totally devoid of any plants. Broken branches may indicate recent activity. When ground has been dug for a grave, existing vegetation is removed. Because of these changes, the type of plants that inhabit that particular area will differ from the established dominant plants in the area. And you could even look at this just at a regular graveyard. If you see a fresh grave versus one that's been there for a couple of years, you'll notice that the new ones are a little bit more mounded up, and then eventually they sink in a little bit. And that is it as far as our presentation on forensic botany. Now, you guys in your notes will have some vocabulary terms to define, so make sure you go through all those. You can look those up online, or you could go back through the notes in the slides if you'd like to. We're going to be continuing on looking at some different case studies next, and I will talk to you guys soon.